Hello and welcome to lecture 8.2, where we'll be talking about the details of the light-dependent reactions. Now, before we get to the content for today, though, let's talk about lecture question 8.1, which the following is false regarding the structures of chloroplast. If you said C, you'd be correct. But why? Any guesses? Do you know why? Well, it's because thylakoids have only one membrane. They don't have an inner and outer membrane. The things that have inner and outer membranes would be, say, the chloroplast itself. There you go. Okay, let's move on to lecture 8.2, the light-dependent reactions. Okay, so light-dependent reactions are kind of the setup for the actual carbon fixation reactions later. So how does this happen? Well, we're going to get some kind of electrons handed off to electron carriers, reducing the NADP plus to NADPH. We'll also make some ATP by having a membrane protein take some inorganic phosphate that's hanging around and stick it onto some ADPs through a process called photophosphorylation. Any guess as to what drives the photophosphorylation process? I'll give you a hint. That's not a trick question. Yeah, it's ultimately photons or light that is driving this process. Now, there's a couple steps in between, but we'll get to that. Now, to make both these things happen, both having electron carriers and ATP, we need some parts, right? Namely hydrogen ions, which will kind of strip down for electrons, or will make them ions for their electrons, right? And then, of course, we're going to need to know where we get that hydrogen. It's from a very readily available source, water. And then what happens when we take hydrogens away from water? What are we left with? Oxygen, yeah. Okay. So let's talk a little more specifics. During the light-dependent reactions, light and energy is converted into chemical energy in the form of ATP and NADPH. Now, we've talked about NAD before, but not NADPH. So what's the difference? NADH versus NADPH. Yeah, it's just a phosphate group, okay? So this process takes place through, I should say, the process of, harnessing light energy to create ATP and NADPH. It takes place through a series of proteins embedded in the thylakoid membrane known as photosystems 1 and photosystems 2. Now, based on lecture 8.1, any guess as to what other type of molecules we'll find in these photosystems? I'll give you a hint. You're going to find proteins for sure, but what else? One more hint. They're involved in capturing light. Yeah, right. Chlorophylls, right? So the pigments that can actually absorb light. So let's take a better look at this system. So the next diagram is going to seem like a lot, but don't sweat it. We'll break it down piece by piece with some beautiful original diagrams created by yours truly. Okay. Now, remember the thylakoids? So that's these structures here inside the chloroplast that look like stacks of green coins. Now, these green coins are called the granum. Don't worry about that so much. Let's just make sure we know what thylakoids are. And they're basically stacks of phospholipid membrane pockets. Okay? This right here is why they are basically stacks and stacks and stacks of membranes. Okay? To maximize the surface area to volume ratio. Why? Well, the surface area is the amount of phospholipid membrane that you have available for serving as a home for these membrane proteins that we're going to be using to do this process. Now, more surface area means more room for more membrane proteins. Okay. Now, figure 8.16 is a pretty simplified kind of cutout of the membrane of the thylakoid with one set of these proteins visible. In these, you'll have the ones dedicated to kind of capturing light, the photosystems 2 and photosystem 1 that you can see here. Notice, annoyingly, photosystem 2, or PS2, actually comes first in the series. The reason we call it PS2 is because photosystem 1 was discovered first. It's kind of dumb, I know, but that's what it is. So photosystem 2 comes first, and then photosystem 1. So that's how they capture the light energy to do some kind of work. Now, they either reduce NADP+, or they're going to be moving hydrogen ions around for later use in ATP synthesis. Okay, so basically, photosystem 2 excites an electron, passes it down the line to get a series of proteins called the electron transport chain. Oops, got ahead of myself here. Basically, photosystem 2 excites an electron and then passes it down the line to a series of proteins called the electron transport chain, or an electron transport chain, where some of this kind of excited energy from that electron is used until it reaches photosystem 1, which then re-excites that and then ultimately passes it off to NADP+, or reductase, yeah. A protein that's, what do you think NADP+ reductase does? Right. Anyway, so this passing of the electron down the line, going from photosystem 2 to the electron transport chain, to photosystem 1, to another very short electron transport chain. Well, this is considered linear electron flow. Now, there are kind of other ways of capturing light energy, specifically cyclical electron flow, but we're going to talk about that later on. Anyway, let's take a closer look at how these different photosystems are structured before we actually start putting all these pieces together. Okay, so there are two major regions in the photosystems. There's the kind of light harvesting complexes that you can see here in the green, and the reaction center that you can see here in blue. The light harvesting complexes are kind of like the solar panels of the operation, filled with a ton of chlorophyll molecules designed to absorb and react with light. Now, there's also these kind of accessory pigments that are there to help pick up some of the stray energy, and there's also proteins there, right, for kind of structural purposes. Now, these light harvesting complexes capture the light energy by exciting a pigment molecule and then pass that energy between pigments until it reaches the reaction center complex. In the reaction center complex are specialized chlorophylls that, rather than simply pass the energy, they're actually oxidized when the energy reaches them. This means the chlorophyll in the reaction center complex can actually pass on an excited electron for use. Now, fundamentally, photosystem 1 and photosystem 2 are very similar in structure and function, but there are a few differences. Structurally, we talked about that already, but on a functional side, PS1 most efficiently absorbs light with a wavelength of about 700 nanometers, while PS2 absorbs light best around 680 nanometers. Because of this, we often refer to PS1 and PS2 as P700 and P680. Okay. let's actually dive into the steps of linear electron flow now. We're going to break it up into 8 different steps, starting with step 1, appropriately. Now, a photon hits the pigments in photosystem 2. And then they kind of pass that energy back and forth through the pigments and the light harvesting complex until it reaches the P680 in the reaction center, which gets the kind of electron excited. Now, step 2 is pretty short. That excited electron is then transferred to a primary electron acceptor, which will pass it on in a minute here. That means P680 has lost an electron. What's the term that's associated with losing an electron? All right. Oxidation is loss. Remember, oil rig, so oxidation is lost, meaning that P680 is oxidized and becomes P680 plus. Okay. Now, before we figure out what happens to the electron the primary acceptor kind of grabbed, right, we need to rectify the fact that we have an oxidized P680 by giving it an electron. In other words, reducing it, right? So we need to reduce P680 plus. Now, where can we get those electrons? Any guesses? If you're not looking at the slide too hard. Right, so we can get those electrons from water. Now, hydrogens are peeled off from the oxygen with their electrons handed to the P680 plus, reducing it back to P680. Of course, water without hydrogen is just an oxygen. So when this process happens a couple of times, we end up with oxygen gas, which is released as a byproduct. Now, step 3 here, where we reduce P680 by splitting hydrogen, basically resets this whole system and lets us repeat steps 1 and 2. Anyway, what about that darn primary electron acceptor that the electron has gone to, right?